Hi there, welcome to Spec Point 2.6, which is protein synthesis, so making proteins. So this is made of two parts, transcription and translation. So let's start off with an overview of transcription and translation and where they're happening. So if this is my cell, and this is the nucleus, in the nucleus of a human, we have 46 chromosomes. Yeah, I'm not trying to draw 46, but we've got 46 chromosomes in there. And those 46 chromosomes make up our whole genome. Okay, so that's all the genes that code for absolutely every protein we could possibly need to make. Now, when we're making a protein, we need to make one protein at a time. There is no way that we're going to do transcription, translation from all of these chromosomes all at once. Okay, so if you remember, when we were to zoom in on a chromosome, sometimes when we draw chromosomes, we draw them with the stripes on them. And each of those stripes represents a different gene. So one chromosome contains loads of different genes. So this gene might be the instructions to make a protein that determines the colour of your eyes. This gene might be the instructions to make a protein that determines the shape of your nose. This gene might be the instructions that codes for a protein that that forms the hemoglobin for your red blood cells, etc. Okay, so we only want one little stripe off one chromosome to be transcribed at a time. All right. Now, if I was to just draw my cell again, the if I zoom in on the nucleus, so if this here is the nucleus, and then this is the rest of my cell. What we're actually going to do in the nucleus is we're going to just copy one gene in transcription. Okay, so from our whole DNA, all our chromosomes, we're going to make a tiny copy, single-stranded copy of just one little gene. So these here are our chromosomes made of DNA. And this little tiny molecule here, which is just a copy of one single gene of one single chromosome, is called mRNA. And that process of making a copy of one little tiny gene from one chromosome, that process is called transcription. So it's making a copy of one gene. And that copy is in the form of this little tiny mRNA molecule. And that transcription happens in the nucleus. Okay. Now that little mRNA molecule, as you can see, is much, much smaller than the DNA was, because the DNA was your whole genome, massive, massive, massive molecules, whereas this is just a copy of one gene. So it's a tiny molecule, and it's so small that it can fit out through these little holes in the nuclear envelope. We call those nuclear pores. Little holes in the nuclear envelope. And the reason why the mRNA can fit out, but the DNA couldn't, is because the mRNA molecule is much, much smaller. Now, this little mRNA molecule then goes out into the cytoplasm and binds on to a ribosome. We draw our ribosomes a bit like snowmen because they've got one small subunit and one large subunit. And our mRNA binds on to our ribosome. So there's our mRNA. And this snowman type thing is our ribosome. Now, normally we could draw those as tiny little dots, either loose in the cytoplasm or on the rough endoplasmic reticulum, but we're zoomed in here to make it a bit bigger. And what happens here is this mRNA is the set of instructions for making a protein. Now, we know that proteins are made out of amino acids, okay? So what happens is these molecules called tRNAs bring amino acids to the ribosome, and the ribosome joins these amino acids together in a long chain that can then fold up to make a protein. And that process there is translation. So translation happens in the cytoplasm or on the ribosome. And what translation does is it uses the mRNA instructions to join amino acids in a specific sequence to make a polypeptide, which folds up into the protein. Okay, so we've got transcription happening over here in the nucleus, and we've got translation happening over here in the cytoplasm. Okay, so let's have a look at transcription in more detail. So transcription is the process where we're going from DNA to mRNA. So how do we use DNA to make mRNA? Okay, so first of all, our DNA is double-stranded, isn't it? So here's our DNA molecule, our double helix structure, DNA. The first thing that happens is we need to unzip that DNA, and we call that a transcription bubble. So you can see here, we've got this area here where it's not bound together. So normally in the DNA, we've got the bases, complementary base pairing of bases, holding the two strands together. So what's happened here is we've broken some of those hydrogen bonds between the bases to unzip this section of DNA. And that little section where it's unzipped is called a transcription bubble. So the unzipping is us breaking the hydrogen bonds between the bases. And we call that bit now a transcription bubble. And what we've then got is exposed bases. So these bases are still there on the nucleotides, but now they're exposed. And we've got two strands. Now we're only going to use one of those strands to make our mRNA. We don't need to make two mRNAs, one of each strand. We're just going to use one strand. And the strand that we're going to use is called the antisense strand, or you can call it the coding strand. All right, that's the one that we're going to use. So what happens now is that we add complementary nucleotides to the antisense strand. So remember that a nucleotide is the phosphate sugar base, yeah, the nucleotide. And we often simplify that shape as a little L shape. So along here, I'm going to add complementary nucleotides to these little bases that are sticking out here. Okay, I've drawn those the wrong way around actually. Let's draw those again. So if these are the bases that are now exposed along here, and we're adding complementary nucleotides like this. So A to T, C to G, except, remember, these blue ones that we're adding in are RNA nucleotides. Okay, we're adding RNA nucleotides. And remember, there is a difference in the bases. So where DNA nucleotides are A, T, C, and G, RNA are A, U, C, and G. Okay, so just be mindful of that. Now, the enzymes involved, when we're unzipping here, the enzyme that does that is called helicase because it's undoing the helix, A's for enzyme. The enzyme that's attaching these complementary nucleotides, let me write that. Complementary RNA nucleotides. The enzyme that catalyzes that is RNA polymerase. Okay, so RNA polymer phase. Okay, so RNA because it's making a strand of RNA. Polymer means a strand of repeating units. So it's look, nucleotide, 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 definitely repeating units. Ase because it's an enzyme. So RNA polymerase is adding these RNA nucleotides using complementary base pairing between the bases. Okay, and the final thing that happens is that they just get joined together. So finally, we just join together the backbone of those nucleotides to form a continuous strand of RNA, and that RNA is called mRNA, which stands for messenger RNA. And that mRNA can then get, um, I might draw the spaces sticking out, will then get released. Sorry, there it is. Our mRNA strand that's made here gets separated and released from the original double-stranded DNA, and that mRNA then continues on its journey. So if we go back to our picture, the mRNA is made and it comes out of the nucleopores into the cytoplasm. So then once it's in the cytoplasm, you can go to a ribosome for translation. So translation is the process when we use the mRNA as our instructions to make a polypeptide. It's the strand of amino acids that folds up to make a protein. It's just called a polypeptide when it's in a line, and then it's called a protein when it's folded up. So our mRNA strand that we've just made is going to go to a ribosome. So the ribosome we draw as a snowman because it's made of a small subunit and large subunit. And the mRNA molecule is going to go in here, almost like the scarf of the snowman. And it's got these little bases sticking out because it's only single-stranded, remember? It's a small single-stranded molecule. So we've got exposed bases here, and the first three, just as one little detail to remember, are always AUG. I always think August, the month August, to help me remember that. Now, in the cytoplasm, we've got lots of these molecules which are called tRNAs. Now, tRNA stands for transfer RNA. Now, another way you could think of it is taxi RNA. They're little taxi services that carry specific amino acids. So these rectangles are amino acids. In most of them, we don't really mind what they are. We could literally just call them amino acid AA to remind us that it's a tRNA molecule carrying an amino acid. Now, on the other end of the tRNA molecule, we've got three little bases sticking out the bottom here. And those three bases there are called the anticodon. So these three bases here, we call the anticodon. And these three bases, or each three bases, so we've got three bases there, three bases there, three bases there, are called the codons. And we need to bind together complementary anticodons and codons. So using complementary base pairing. So A will bind to U because it's not DNA, it's not T. And then C's will always bind to G's. We call that complementary base pairing. So it's literally trial and error. So they might try this one. Is the anticodon complementary? No, let's try this one. Is the anticodon complementary? No, let's try this one. Until the right one binds. Now, whatever this anticodon is, the amino acid that it carries is very specific to that anticodon. So the first tRNA that always binds, it's always the same. Hang on, I'm not going to fit it in there. Let me write the letters first. I'm going to have the complementary anticodon to this. So A will bind to U, U binds to A, G binds to C. Okay, so the first tRNA that binds has the anticodon UAC, complementary to AUG. And it always carries the amino acid MET, methionine. Okay. And that's always the first one. So this codon here, the first one, is called the start codon, and it binds to the complementary tRNA, which is carrying the amino acid methionine. And then what will happen is that this ribosome here will actually move along, and we'll get another tRNA bind in this space here. So the next tRNA comes along and it binds to these three, and it carries with it another amino acid. And what happens is that these amino acids at the top here join together, just as we've learnt amino acids join together in topic 1, they join together with a peptide bond. Okay. Now, interestingly, this first tRNA actually gets reused. So that tRNA will detach, but it will leave its amino acid in the chain just there. Okay. Then what happens is, as we said before, the ribosome moves along. Let's just put this on a little bit. So the ribosome then moves along. So let's just put it just here for now. And another tRNA comes along. So the next tRNA is going to bind just here with the next amino acid. A peptide bond will form in between the two amino acids. And then the next tRNA will leave the ribosome, and it will leave behind its amino acid. You're starting to get the idea. And another tRNA will come in. So this mRNA, I didn't draw it very big, but really this mRNA is going to be long, isn't it? So then it can bind to the next 3 bases, and the next 3 bases, and the next 3 bases, and it builds up this chain of amino acids. And a chain of amino acids is called a polypeptide. So it will do that all the way along until it gets to the end of the mRNA. And once it's finished, this will fold up. Remember, primary structure is the sequence of amino acids, and that's how we get the specific sequence. Secondary structure, it will wind up into alpha helices in some bits, beta pleated sheets in other bits. Tertiary structure, overall 3D shape. Possibly quaternary structure, where one polypeptide joins with another polypeptide, possibly even more than 2 together. So it folds up into a protein. Okay, let's go through the steps of translation in writing, just to help you with your notes as well, because I've done that very much in pictures. So first of all, what happens is the mRNA binds to the ribosome, and the ribosome is in the cytoplasm. Then what happens is that a tRNA with a codon that is complementary to the first codon of the mRNA will bind. Okay, so you've always got to mention that's anticodon there, that the tRNA has anticodons and the mRNA has codons. So you've got to get all those words in. So the tRNA with an anticodon that's complementary to the first codon of the mRNA will bind using complementary base pairing. Now, they always want you, when you say complementary base pairing, to say what the pairs are. So because we're talking about RNA here, not DNA, it's not A and T, it's A and U and C and G. Okay. Next, what happens is this repeats and another tRNA binds. Then what happens is the two amino acids held at the other end of the tRNA bind together by a peptide bond. The first tRNA then leaves the ribosome to pick up another amino acid, leaving the first amino acid attached to the growing polypeptide. Another tRNA binds, bringing another amino acid. The amino acid joins to the chain. The tRNA leaves, and it repeats over and over and over again until you've got your finished polypeptide, and finally the polypeptide folds into a protein. Alright, and that's how we make a protein. So going back, just to summarise, we always start off in the nucleus, but we don't want to make the proteins of all the chromosomes at once, all the genes at once. We're just making an mRNA of one single gene. And the mRNA that we make is small enough to fit out through the nuclear pore, go to the ribosome, where it's used as a set of instructions to join together amino acids to make a protein. And then we've gone through the details of how the DNA is used as instructions to make an mRNA, and then translation, how the mRNA is used as instructions to make the polypeptide. Okay, that's the end.